Good morning, I'm David Smith, and today I'll share some work from my PhD about how microbiome-pathogen interactions influence the epidemiological dynamics of antibiotic resistance. Antibiotic resistance is a problem that bridges scales, from resistance mutations in individual cells, to colonization dynamics within the host, to patterns of host-to-host transmission, to the global spatiotemporal distributions of drug-resistant organisms. And Eve Rabé will give a nice presentation on this last topic later in the session. In light of this complexity, we still have an incomplete understanding of how antibiotics select for antibiotic resistance across these different levels of organization. In this talk, I'll focus on the host population level and how antibiotics select for the epidemiological spread of resistance in healthcare settings. And in particular, I'll focus on how these between-host dynamics are influenced by within-host interactions with other bacteria, which is a leading area of study in the epidemiology of antibiotic resistance. It is believed that competition between drug-sensitive and drug-resistant bacteria drives how antibiotics select for resistance. The reasoning goes as follows. If drug-sensitive cells CS and drug-resistant cells CR are in competition with one another, then antibiotics that preferentially clear sensitive cells provide a competitive advantage to resistant cells, making them more likely to reproduce and transmit. Building this within-host competition into a between-host epidemiological model offers a mechanism by which increasing antibiotic use favors the spread of drug-resistant bacteria. This mechanism can also be used to explain selection trade-offs, as seen in this illustration, where an intermediate level of antibiotic exposure maximizes selection for resistance. Yet recent reviews suggest that we lack a complete understanding of key principles, including how much drug-sensitive and drug-resistant strains interact and compete, what causes antibiotic selection trade-offs, and how microbiome-pathogen interactions influence epidemiological dynamics. Indeed, most models consider just two competing strains of a particular bacterial pathogen, yet in reality, bacteria that colonize the human body live in complex polymicrobial communities. This is particularly important in the context of antibiotic use because antibiotics can destabilize the microbiome, causing a state of disequilibrium known as dysbiosis, which increases risk of colonization with drug-resistant bacteria. Further, eventual repair of microbiome stability may help control and eliminate drug-resistant species. In this talk, I'll present a novel modeling framework that accounts for impacts of within-host ecological interactions on the epidemiology of antibiotic resistance. Then I'll apply this framework to simulate the public health efficacy of a range of hospital infection control interventions. The main objective of this work was to develop models of bacterial colonization in healthcare settings, where each model describes distinct within-host ecological interactions between a focal bacterial pathogen CR and other bacteria. Because of time limitations, I'll quickly move through the 5 models that we considered. The first includes no within-host interactions. The second includes intraspecific strain competition. The third includes competition between the focal pathogen and the host microbiome. The fourth includes both strain competition and microbiome competition. And finally, model 5 includes horizontal gene transfer of a focal resistance gene. Our approach was to simulate pathogens over a wide parameter space across all models to evaluate impacts of these diverse interactions on colonization dynamics. In the first model, we make a number of standard assumptions. First, we include multiple routes of acquisition, including a rate of host-to-host transmission determined by the dynamic force of infection lambda, and a rate of endogenous acquisition alpha. Second, we include multiple routes of pathogen clearance, including a rate of natural clearance gamma and a rate of antibiotic-induced clearance sigma. And finally, we include host demography with a constant population size and steady introduction of the pathogen into the hospital from the community. All forthcoming models build on these assumptions. The second model is an exclusive colonization model, which adds a second strain CS to the previous model, assuming that hosts can be colonized by either one strain or the other. This is the most widely used strain competition model in the literature. However, we propose a novel means of modeling antibiotic clearance of each pathogen strain J. Here, antibiotic-induced pathogen clearance depends on the exposure prevalence of antibiotic treatment A, the share of antibiotics against which the pathogen is resistant, R, and the clearance rate due to effective treatment, theta C. We model antibiotics this way for 2 main reasons. First, this formula allows us to decouple antibiotic exposure from antibiotic-induced colonization clearance, which is important later when we consider other impacts of antibiotics. Second, this formula results in an antibiotic selection trade-off for any strain K having a higher resistance proportion R than the competing strain J. We can visualize this on the next slide, where here on the x-axis is antibiotic exposure prevalence, ranging from 0 to 100% of patients, and on the y-axis is pathogen colonization prevalence at population dynamic equilibrium. In this case, prevalence of the more drug-sensitive strain CS always declines with increasing antibiotic use, while the more drug-resistant strain CR is bounded by a trade-off. At low antibiotic use, antibiotics tend to favor CR by preferentially clearing CS and creating more opportunities for CR colonization, but at high antibiotic use, antibiotics tend to limit both strains. Moving on from this more traditional modeling framework, we developed a new model that instead accounts for microbiome-pathogen interactions. This model describes colonization with a single pathogen strain, CR, and assumes that hosts can have either a stable microbiome or can undergo antibiotic-induced microbiome dysbiosis. We assume that dysbiosis occurs at rate sigma m, which also depends on antibiotic exposure prevalence A, as well as the rate at which patients exposed to antibiotics undergo dysbiosis, theta m. The key hypothesis we build into the model is that patients with a stable microbiome are more resilient to pathogen colonization, while hosts undergoing dysbiosis are more susceptible. And this is modeled through three different ecological interactions: colonization resistance, resource competition, and ecological release. I'll zoom in now to the within host level to demonstrate how these interactions were conceptualized and modeled. Here's an illustration of the host microbiome at healthy equilibrium on the left and undergoing antibiotic-induced dysbiosis on the right. We assume that hosts at equilibrium benefit from colonization resistance, in which microbiota inhibit transmitted pathogen cells from establishing colonies. Conversely, among patients undergoing dysbiosis, loss of microbiota reduces within host competition and facilitates pathogen persistence. Lastly, patients undergoing dysbiosis may also experience ecological release, whereby subdominant pathogens are more likely to emerge endogenously due to reduced microbiome competition. On the right here, we evaluate the impacts of microbiome-pathogen competition on pathogen colonization dynamics. Each color represents a different interaction considered in the model, demonstrating that each interaction can independently explain how increasing antibiotic use favors the spread of the drug-resistant pathogen CR, and why intermediate antibiotic use maximizes its spread. Here, this trade-off owes to the fact that antibiotics clear CR colonization while increasing host susceptibility to colonization at the same time. Fourth, we consider a model that combines strain and microbiome competition. In other words, strains CS and CR of the pathogen species compete for hosts, as in Model 2, but host microbiota also influence susceptibility to colonization, as in Model 3. Here, the leading assumption is that microbiota interact equally with each strain. For this model, outcomes vary depending on how antibiotics affect different types of bacteria. This plot here visualizes two epidemiological outcomes at once. The circle size describes the prevalence of the focal pathogen, and the color scale describes the resistance rate, or the proportion of strains that are antibiotic resistant. Along the x-axis is an increasing impact of antibiotics on pathogen strains, and along the y-axis is an increasing impact of antibiotics on the microbiome. Overall, antibiotics that disrupt both microbiota and sensitive strains select more strongly for resistance than antibiotics targeting only one or the other, and this can be seen in the top right corner. Further, there is an asymmetry across epidemiological outcomes. Antibiotics that target sensitive strains lead to an increase in resistance rates, but have limited impact on total pathogen prevalence, while conversely, antibiotics that target microbiota lead to an increase in pathogen prevalence with little impact on resistance rates. Finally, in Model 5, we include interspecific horizontal transfer of a resistance gene R between host microbiota and the focal pathogen. We make a number of simplifying assumptions. Most importantly, the resistance gene bears more resistance to antibiotics, but bears a metabolic cost. This plot is similar to ones we've seen previously, with antibiotic exposure prevalence on the x-axis and colonization prevalence of the drug-resistant strain on the y-axis. And here, dashed and dotted lines represent simulations with HGT. As expected, HGT increases prevalence of the resistant pathogen. However, we also find that HGT introduces a third antibiotic selection trade-off, where the impact of horizontal gene transfer on resistance prevalence is maximized at intermediate antibiotic use. This is because at low antibiotic use, there is limited selection for the resistance gene, while at high antibiotic use, most potential HGT recipients are cleared by antibiotics. Our next goal was to apply this modeling framework to evaluate impacts of microbiome dysbiosis on nosocomial infection control. And due to time limitations, I'll only very briefly introduce this second part of our study. First, we simulated the transmission dynamics of 4 high-risk pathogens in the hospital setting. Second, we simulated a range of public health interventions. And finally, we estimated the efficacy of these interventions for preventing nosocomial pathogen acquisition. Here, this plot shows the efficacy of different interventions over increasing levels of intervention compliance on the x-axis. First, for C. difficile, we found that microbiome recovery therapy was the most effective intervention. For MRSA, traditional contact precautions were most effective. And for the 2 enterobacteria considered, microbiome recovery therapy and antibiotic stewardship were both highly effective, while contact precautions had limited impact. In conclusion, this work represents a novel modeling framework describing the impacts of diverse within-host bacterial interactions on the epidemiological spread of antibiotic resistance. We found that distinct interactions underlie distinct antibiotic selection trade-offs, each offering different explanations for why intermediate antibiotic use maximizes resistance. We also found that distinct interactions underlie heterogeneous impacts on epidemiological indicators, with strain competition explaining how antibiotics drive increased resistance rates, but with microbiome competition explaining how antibiotics drive increased colonization incidence, Finally, this work demonstrates that within-host interactions may have important consequences for the efficacy of public health interventions. In particular, findings suggest that efficacy of antibiotic stewardship for colonization prevention may primarily be due to averting dysbiosis. Further, microbiome recovery therapies may be promising tools to control resistance dissemination, although further development of these interventions and future trials are needed. There were also a number of limitations in this work. For instance, we did not account for stochasticity, and there is a lack of longitudinal clinical data needed to robustly inform some model parameters. So that's it for me. If you'd like to read more about this work, please don't hesitate to check out our paper recently published in eLife. And thank you very much for your attention, and I'd be very happy to take any questions.